Hi everyone, my name is Marko Lukic. I work as a curator at the Croatian Natural History Museum, and today, together with my colleague Rožman, I will present the discovery of a novel adaptation to life in caves, the controlled jumping behavior in Collembola. Let me begin with an introduction to Collembola and the caves they inhabit. Collembola are small six-legged arthropods, usually only 1 to 5 millimeters long. They are extremely abundant and occur in almost every terrestrial and semiaquatic ecosystem, and they are also very common and diverse in caves. Collembola are also called springtails because they possess a specialized catapult-like organ called furca. This organ allows them to jump explosively and high into the air. Springtails use these jumps primarily to escape predators, although a few species use them for migration. For a long time it was assumed that these jumps were completely uncontrolled. However, recent studies have revealed controlled jumps in one semi-aquatic species. Videos filmed with ultra-high-speed cameras reveal the true magnitude of their uncontrolled jumps. Takeoff occurs in just 1.7 milliseconds, and the animals can rotate uncontrollably at more than 350 turns per second. These takeoffs were filmed at 50,000 frames per second. Caves are other extreme habitats. They are permanently dark, nutrient-poor, and with high humidity. Animals that colonize caves often exhibit distinctive adaptations such as loss of pigmentation in eyes, elongated appendages, and behavioral changes like increased activity, altered circadian rhythms, or modified feeding strategies. Most of these behavioral studies have been done on cave vertebrates such as fish or salamanders. Collembola also exhibit many morphological adaptations to life in caves. Today I will emphasize the modification relevant to this study, the elongated claws that you can see here on the image, which improve grip on wet and slippery surfaces. Claw elongation is almost universal among cave-dwelling Collembola, suggesting that wet substrates are important pressure-affective movement in these tiny arthropods. During fieldwork in a Croatian cave, we observed a novel adaptation in Collembola. The habitat where we filmed this behavior consists of a series of small sinter pools filled with water, and this is a major obstacle for animals the size of a sesame seed that you can see here. Here's one specimen of Collembola. The microendemic species of genus Isotomurus performs controlled jumps over these pools. Unlike typical springtail jumps, these were horizontally directed and adjusted in strength, allowing precise control landing. Instead, the tip moves on the edges of these small sinter pools that is longer way around. It reaches the water and jumps across the small pool. The landings are not always successful, but the springtails are persistent and maintain their movement direction even after unsuccessful attempts. It also uses small jumps in order to overcome these small vertical obstacles. Although we do not have high-speed cameras available, a normal slow-motion footage that you will see in a few seconds illustrate the precision of their landing. The same individuals were also capable of typical uncontrolled jumps when disturbed, suggesting that controlled jumping is used for navigation rather than escape from predators. This behavior was confirmed on several occasions, and we are conducting further tests in custom-built arenas with closely and distantly related cave and non-cave dwelling Collembola to confirm our discovery. In conclusion, controlled jumping in cave habitats, together with claw elongation, likely facilitates movement across constantly wet and periodically flooded cave floors. This highlights how morphological and behavioral traits can work together to overcome the challenges of locomotion in cave habitats. To our knowledge, this is the first documented behavioral adaptation of cave-dwelling Collembola and a rare behavioral adaptation among cave-dwelling arthropods in general. I would like to thank the Croatian Biospeleological Society and its members for the help during fieldwork and filming. This research was supported by the Systematic Research Fund and the Linnean Society of London and the systematic association. Thank you very much for your attention, and I will be happy to take questions. Thank you.